Good morning. Dr. Morris here today to talk about the physiology of the gastrointestinal system. It's been a while since I've recorded one of these videos. Today I decided to just wear the t-shirt that I normally wear in quarantine instead of dressing up and pretending I'm at the office. On the other hand, I want to emphasize that if you were to ask Dr. Christina Perry, our program director, you are about to begin the most important module in the physiology course. So, with that in mind, I would like to say that this material was developed by both me and Dr. Perry, so her input is very much appreciated in these next two PowerPoints. This is the objectives for today's lecture. We're going to start by talking about some general things about the gastrointestinal system, including the parts and the functions and activities and a little bit about the anatomy, moving into discussions of how the activities of the GI system are controlled, including neural signaling and endocrine signaling, with some emphasis on the fact that unlike the rest of the organ systems that we're talking about, the gastrointestinal system has its own nervous system called the enteric nervous system, sometimes called the little brain, because it is independent in some ways of the central nervous system. It is independent of the peripheral nervous system, although it receives input from the autonomic nervous system. And then we will move into a kind of journey through the alimentary system, starting at the mouth, and in this lecture ending at the stomach. We'll pick up in the next lecture in the small intestine, moving through the anus, and then we'll talk about some of the organs that are accessory organs that are attached to and part of the gastrointestinal system, while not being the hollow visci, not being the tube through which food moves to be processed and absorbed and ultimately have the waste products eliminated. So that's what we're going to be doing in these next two lectures. Let's start by talking about the activities of the alimentary tract, which is a set of structures and organs that does some very important things. The main thing that it does is to take food, break it down into absorbable components, and then, which is called digestion, and then absorb those nutrients into the bloodstream so that they can be used by all of the other organisms in the body. And this occurs through several activities. Food has to be moved through the alimentary tract, and we will talk about the propulsion of food, how it is ingested, mixed with secretions, and then moved by smooth muscle through mechanisms that we call motility from the mouth, swallowing into the esophagus, and then throughout the gastrointestinal tract until it is eliminated as waste products at the rectum, or from the rectum at the anus. There is an activity of the alimentary tract that we call secretion. So from the mouth, where salivary glands are adding water and enzymes and mucus to the food that is being chewed, throughout the rest of the gastrointestinal tract, substances including water and electrolytes, acid and base, and enzymes are being secreted into the food and chyme mixture to allow breakdown and digestion of the nutrients and to promote absorption. The next activity is absorption. So through the course of parts of the alimentary tract, starting at the small intestine, water is absorbed. Many electrolytes, and including things like calcium, are absorbed. Vitamins and other minerals and essential nutrients are absorbed. And then the products of digestion, the breakdown products of carbohydrate, protein, and fat are absorbed. And that's an important activity, and we'll talk about it in a good deal more detail. There is blood circulating throughout the gastrointestinal tract, crucially, not just to provide oxygenation to these very metabolically active cells, but more importantly, perhaps, or as importantly, to pick up the products of digestion and to carry them away. And then all of these activities are controlled by various systems, including local factors that include things like paracrines and neurocrines, substances being emitted to act on cells in the vicinity. Neural signaling, which is both from the autonomic nervous system, sent from the central nervous system to the peripheral nervous system through the autonomic ganglia, but also the enteric nervous system, which is unique to the gastrointestinal system. And then finally, hormones. And just as we have talked about other organs that emit hormones that we don't think of as part of the endocrine system, for instance, the heart, which is sending out atrial and brain BNP, natriuretic peptide, and the kidney, which, as we've talked about, makes renin. The alimentary system makes a number of hormones which are absorbed into circulation and act at a distance from the cells that make them. The alimentary tract starts at the mouth, includes the salivary glands, proceeds through the pharynx to the esophagus, the stomach, and then from the stomach with inputs from the pancreas, liver, and gallbladder through the small intestine that consists of the duodenum, the jejunum, and the ileum, and then through the ileocecal valve into the ascending colon, the transverse colon, and the descending colon, where the feces, the waste products that remain, are stored in the rectum until they are eliminated through the anus. So those are the components of the alimentary tract. I want to talk a little bit about some general features of the anatomy of the wall of the GI tract. So from the esophagus through the rectum, these layers exist. I would like to say that there is an exception, and we will get to that exception where there is an additional layer. But for now, know that the walls of particularly the intestine consist of at least 5 layers. Those layers include the serosa, which is the outer membrane facing out into the peritoneal cavity. It is where blood flow comes in, and it surrounds all of the tissues of the intestine and the stomach, and is continuous with the mesentery, which is the structure through which blood flow comes to the intestines. Interior to the serosa, there are 2 layers of muscle. There's a longitudinal layer that is just immediately interior to the serosa, and a circular layer that is interior to the longitudinal layer. Under the circular layer is something called the submucosa, which provides the basis on which the mucosal epithelial layer resides. Now, it's important to know that there are nerves and neurons that exist in the layers of the GI wall. Between the two layers of muscle, the longitudinal and circular layer, is a plexus, a collection of neurons and nerve fibers called the myenteric plexus. Between the inner layer of muscle, the circular muscle, and the submucosa exists a second plexus that is called the submucosal plexus. These have other names, and we will show you those in a minute. The submucosal plexus is also called Meissner's plexus, and the myenteric plexus, is also called Auerbach's plexus. But I think that the names myenteric, which kind of implies it's associated with muscle, and submucosal, that it's associated with a submucosal layer, are more descriptive and more useful for your memory. I wouldn't ask you a question about Meissner's plexus without further categorizing it as the submucosal plexus. And these neurons and nerve fibers are the enteric nervous system that consists entirely within the tract, the walls of the GI tract. Here is another picture showing exactly the same thing. So this time let's move from the inside out. We'll start with the mucosa, which varies through the gastrointestinal tract. It has different functions and different structures that we'll talk about as we move through the tract. But this inner layer is a mucous membrane, an epithelium, a single layer. Within that, there are glands. So here we have some submucosal glands, and these are glands that are emitting things into the lumen, and this is the lumen of the gut. There is a submucosal layer around the mucosa, supporting the mucosa. There are two layers of muscle outside of the submucosa, and those layers of muscle include a circular muscle that is closer to the lumen and longitudinal muscle that is closer to the outside. And then there's this single layer of tissue surrounding the entire structure called the serosa that is continuous with the mesentery. And then remember that there are those two neural plexuses that are part of the enteric nervous system or little brain. There is the myenteric plexus that's between the two layers of muscle, and there is the submucosal plexus that is within the submucosal layer, or between the submucosal layer and the circular layer of muscle. I'd like to talk a little bit more about the enteric nervous system and how it controls function in the gastrointestinal tract. So again, this enteric nervous system is entirely within the wall of the gastrointestinal system. It starts in the esophagus, it ends at the anus, and it is composed of two sets of neural bodies and fibers that we call plexuses. The myenteric plexus that's between the longitudinal and circular muscle layers, and functionally that is the layer that primarily controls movement in the gastrointestinal tract, but also relaxation of circular muscle at certain points, or relaxation or tonic contraction. So the pylorus, which is the area of the stomach that is sort of a valve from the stomach to the small intestine, and the ileocecal valve, which is an area that allows passage of food from the small intestine to the ascending colon. So movement, muscle function is controlled by the myenteric plexus. The submucosal plexus, that lies in that submucosa is primarily responsible for controlling blood flow and also controlling the secretion of all those things we talked about that get secreted into the lumen. So water and acid and alkaline, bicarb, and digestive enzymes of many sorts. It's important to understand that while we have this independent enteric nervous system, that is able to function independently in the same way that the heart pacemakers can function independently of neural input. There is neural input, just as there is normally to the pacemaker of the heart, and that is autonomic input, both sympathetic and parasympathetic. If you remember from our lectures about the autonomic nervous system, the sympathetic ganglia are far from organs, and so these fibers are postganglionic fibers, but the parasympathetic ganglia are close to and within the organs, and so there are actually preganglionic parasympathetic fibers going to ganglia in the enteric nervous system. So we get input from the sympathetic system, we get input from the parasympathetic system, but we also locally have receptors that detect stretch in the muscle. They are mechanoreceptors, and there are chemoreceptors that detect things like pH, but also the presence of certain products of digestion, like amino acids and fatty acids. And those are within the gut mucosa, and they send signals that can act locally, both paracrine hormone-like signals that act in the vicinity of where they're released, and neural signals, neurocrines, which are neurotransmitters that act locally. And finally, there are sensory neurons that, and we mentioned the mechano and chemoreceptors that act on the enteric nervous system, but there are also sensory neurons that go to the spinal cord and brainstem and help to regulate the sympathetic and parasympathetic signals that are coming back. There continues to be much investigation about the types of neurotransmitters that are used in the enteric nervous system. And while the major ones are acetylcholine and norepinephrine, there are many others that include things like serotonin and dopamine, substance P, nitric oxide, and not all of the functions are known, and as I said, investigation continues. But be aware that this is at least partly why the gut is as influenced by emotion and nervous system issues as it is. Again, we have not yet a full understanding of this, but it's an important thing in clinical practice. So acetylcholine, which is the same acetylcholine that's used by the autonomic nervous system at all the preganglionic fibers and at the postganglionic fibers of the parasympathetic system, primarily excites GI activity. It speeds things up, whereas norepinephrine tends to inhibit activity in the GI tract. And in both cases, that is both motor and secretory activity. There are also a number of peptides that are secreted within the GI system. Some of them are hormones, and an example is gastrin, which is a hormone secreted by cells in the stomach wall lining, but that rather than being secreted into the lumen are secreted into the capillary circulation and act at a distance to have effects on the GI tract. There are neurocrines, which are substances that are released after an action potential and act at a synapse on a target cell, and that target cell could be a smooth muscle cell, a neuron, or another cell. an endocrine cell, for instance. There are paracrines that act locally on target cells, so in the vicinity of the cell that releases them. And paracrines are hormone-like chemical messengers, as we discussed early in the course. But there are still many others, some identified, some not, some with known functions, some not. As we go through this lecture, I'll tell you of the ones that I really want you to pay attention to. So there are reflexes within the nervous system of the GI tract. And there are three types of reflexes that are anatomically a little bit different. There are reflexes that are completely within the gut wall, that are completely within the enteric nervous system. And they include reflexes that control things like GI secretion into the lumen, peristalsis, and mixing contractions, and inhibitory effects locally. There are reflexes that start in the gut with a sensory neuron, sending signals to the prevertebral sympathetic ganglia and then signals back out to the GI tract. And in this case, some of these can act over a long distance in the GI tract rather than nearby. So an example of this is that when you eat, there's a signal from the stomach that ends up leading to the mass movements that cause you to evacuate your bowels. So it's called the gastrocolic reflex. And then there are reflexes from the gut that go to the spinal cord or to the brainstem. So the number two reflexes are acting within the sympathetic system or using the sympathetic system along with the enteric nervous system. And the ones in number three are parasympathetic, right? Because spinal cord or brainstem. And again, these include many reflexes that involve motor activity, secretory activity, pain reflexes. When pain sensors in the gut are stimulated, there is a decrease in parasympathetic output that causes general inhibition throughout the GI tract. And then there are reflexes from the colon and rectum to the spinal cord and back that help to produce the necessary contractions to result in defecation related to stretch. There are lots of gastrointestinal reflexes. These are the three types. This chart I like because it shows how there are both short and long reflexes in the digestive system, and there are stimuli that are external and there are stimuli that are internal and local. External stimuli include things like the sight of food, the thought of food, the smell of food, that act at sensory receptors or the brain. Initially thought of food, but sensory receptors to the brain, and then send signals through the autonomic nervous system to the enteric nervous system, where they act on either smooth muscle or cells that secrete substances into the lumen, resulting in either muscle contraction or relaxation or exocrine secretion into the lumen of substances like enzymes and water and bicarb and acid, also on cells that will release things locally that are called paracrines, and in some cases acting on endocrine cells in the GI tract. Local stimuli include things like acidity, stretch from mechanoreceptors, osmolarity, and various products of breakdown of foodstuffs into smaller molecules that are detected by chemoreceptors. And again, there are sensory receptors and neurons in the mucosa. Well, there are sensory receptors in the mucosa with neurons and interneurons in the enteric nervous system, acting again on specific neurons in either the myenteric or submucosal or both plexuses that then act on smooth muscle and secretory cell effectors that then cause responses. So this is a nice little chart, color coded, showing you some short reflexes that act locally and some long reflexes that involve the central nervous system. I'm moving on from neural control to blood flow in the gastrointestinal system. And the circulation in the gastrointestinal system is a little different than it is in some other organs. It is called the splanchnic circulation, and the splanchnic circulation includes the intestine and then the spleen, the pancreas, and the liver, although it also has some input to the stomach. One of the things that is a constant in physiology is that local blood flow tends to be related or proportional to local need for blood flow. So there are local factors that change blood flow. When we are absorbing nutrients, we need more blood flow to the villi and the submucosa to pick up those nutrients. And this is true. I don't know if you remember, when the oxygen need in the cardiac muscle is higher, the demand is higher and blood flow is higher. And we get angina when we're not able to keep up with that demand because there are restrictions to the blood flow. So during GI activity, there are vasodilator substances that are released from the mucosal cells. There are glands in the GI tract that release kinins, which are substances that are vasodilators that often act locally. And then we have noticed that when there is a decrease in oxygen concentration in the gut wall, that increases blood flow because of increased demand. There are cases of people who have severe peripheral arterial disease. We call these folks vasculopaths, who, when they eat, because they can't meet demand, get bowel ischemia and pain, and they develop, because of that, a fear of eating that's called cytophobia. So this is a known response to bowel ischemia that results from increased demand because of local factors, because of the need to absorb food. So therefore trying to dilate vessels that will not be allowed to dilate because of fixed obstruction. So people with kind of mesenteric ischemia that is acting a little bit like angina, that increased demand trying to increase flow results in ischemia, resulting in pain, and in this case resulting in weight loss because of the inability to eat. And this diagram shows something extremely important. So the arterial supply is divided in, there are three main arteries that supply the gastrointestinal tract: the celiac artery, the superior mesenteric artery, and the inferior mesenteric artery. Intestines are supplied by the superior and inferior mesenteric artery. Stomach, spleen, and pancreas primarily by the celiac artery. And there's a hepatic artery off the celiac artery that goes to the liver. On the other hand, all of the venous blood flow out of the gastrointestinal organs enters the portal vein. And the portal vein carries all of this back to the liver and through the liver into the sinusoids of the liver, allowing the nutrients that are absorbed in the GI tract, along with various hormonal substances that are released from organs in the GI tract, to enter the liver and be processed in the liver. So, for instance, when we're absorbing glucose from the small intestines, that all that glucose has to go through the liver. The insulin that is being secreted from the pancreas also all ends up in the liver. Cells in the liver are going to respond to the influx of glucose and insulin to create glycogen, which is that carbohydrate storage product that we talked about in the endocrinology lecture about the pancreas. So all the blood flow goes through the portal vein, through the liver, and then out of the liver from the hepatic veins into the inferior vena cava. Now we're going to move on to hormonal control of GI motility and secretion. So we've talked a good deal about neural control. We mentioned blood flow. And blood flow is important when we're talking about hormonal control. I'm going to talk about a number of hormones that are secreted by the GI tract. The two that I'm going to suggest you learn in detail are on this slide. The first is a substance called gastrin. It is secreted by G cells in the antrum of the stomach. The G cells are in the mucous membrane of the stomach, in the pits and gastric glands. But rather than being emitted into the lumen of the stomach, this substance comes out of the basal side of the mucosal cells into the extracellular compartment and are picked up by capillaries and travel to other areas within the GI tract. Gastrin is important for stimulating the secretion of hydrochloric acid in the stomach. So when signals trigger the release of gastrin, then gastric acid secretion is triggered. In addition, gastrin is a growth factor for the gastric mucosa. The gastric mucosa takes a lot of insults, both physical insults because of friction, because of food going through and all of the mixing and motion, along with chemical insults from certain digestive enzymes like pepsin and, of course, hydrochloric acid. So gastrin acts as a growth hormone. CCK is a hormone you'll hear about a good deal, cholecystokinin, and it's secreted by cells in the mucosa of the duodenum and jejunum in response to breakdown products of nutrients that are coming through the early small intestine. So fat and fatty acids and monoglycerides trigger the release of cholecystokinin. Cholecystokinin then stimulates the pancreas to secrete digestive enzymes into the lumen. It causes the pancreas to increase bicarbonate secretion, and it triggers contraction of the gallbladder, which then pushes bile out of the gallbladder through the common bile duct into the pancreatic duct and out of the sphincter of Oddi. Cholecystokinin also inhibits gastric emptying, and we'll talk about that a little bit more when we get to the stomach. But when food starts making it, fatty food starts making it to the first part of the intestine, the gastric emptying is slowed down in order to control the amount of chyme that is being pushed out of the stomach into the small intestine. Other hormones that are involved in the control of GI motility and secretion include secretin, which is secreted from cells in the mucosa of the duodenum called S cells that stimulate stomach cells to make pepsin, that stimulate the pancreas to make more bicarbonate solution. And bicarbonate's really important in the duodenum because we have this very low pH chyme coming out of the stomach and it needs to be neutralized. The stomach is made to handle low pH. The duodenum isn't. It also stimulates bicarbonate secretion from the biliary ducts, and it tends to inhibit gastric acid secretion. Then there are gastric inhibitory peptide, GIP, which is also called glucose-dependent insulinotropic peptide that stimulates insulin release in response to fatty acids and amino acids in the upper small intestine. It, again, recognizing these nutrients coming through, tends to slow gastric emptying, and it inhibits gastric acid secretion. And finally, motilin, which is a substance that is secreted by the stomach and the upper duodenum when we're not eating, when we're fasting, and that increases GI motility, causing these waves of increased peristalsis and GI motility about every hour and a half when you haven't eaten, and are probably part of the hunger mechanism. And here is just all of that on a slide that kind of summarizes it. So gastrin, secretin, CCK, cholecystokinin, and GIP. So speaking about GI motility, the smooth muscle in the gastrointestinal tract, all the layers, function as a syncytium. So the muscle cells are arranged into fibers, which are arranged into bundles. Some of them are longitudinal, some of them are circular. Longitudinal extends along the length of the gastrointestinal tract, and circular is in a circular arrangement around the gut. And between those fibers, there are gap junctions that allow for the transmission of action potentials. These cells are electrically connected, and so when one cell has an action potential and begins to contract by electrical chemical excitation, the adjacent fibers also contract. There is some connection between the longitudinal fibers and the circular fibers as well, so they can work together. As longitudinal fibers are excited, the circular fibers can also join in, and these impulses travel along lengths of the intestine. There are factors that make the cell membranes of those smooth muscle cells less stable, more excitable. They include stretch, parasympathetic input, or acetylcholine stimulation, and certain GI hormones can make those muscles more likely to contract, to be excitable. And there are similarly factors that make those muscle fibers less excitable. They include the effects of epinephrine and norepinephrine. Norepinephrine from the sympathetic nerve fibers, epinephrine that's circulating. So when the sympathetic nervous system is triggered, the gut tends to slow down because the smooth muscle is less likely to be motile. There are roughly two kinds of functional movements in the GI tract. Functional meaning that they're performing a specific function or activity that's important. There are propulsive movements, which we also call peristalsis, in which a area of smooth circular muscle around the gut contracts, and then that contraction moves forward in a direction primarily from mouth towards anus, and the material in front of that ring as it moves is pushed forward. The usual trigger for peristaltic movement is distension. So an area of the gut is distended and a circular ring forms, which then propels the distended area forward. Peristalsis can reverse, but in general, there is a polarization of that myenteric plexus in the direction of the anus, and so most peristaltic movement is from the mouth towards the anus. There are also something called mixing movements, and in general, there's a lot of mixing just from peristalsis, but in some parts of the intestine, particularly the small intestine, there are these local circular muscle constrictions every few centimeters that then relax with the next areas of constriction in different points. So this results in chopping up of the chyme of the contents and then continues its chopping and mixing. Peristalsis, on the other hand, typically is a constriction behind a bolus that then moves the bolus forward with subsequent relaxation of the muscle ahead of the bolus. We begin mixing and propelling our food in the mouth. There is chewing, clearly, which is both grinding and mixing food in the mouth. The motion of swallowing is the beginning of propulsion of food out of the pharynx into the esophagus. There is peristalsis in the esophagus. There are a number of motions that occur in the stomach, which both mix food but also slowly propel it forward as needed. And then the small intestine, and then the large intestine. So as we move through the different areas, we will talk about how food is both propelled and mixed in those areas of the gastrointestinal system. I want to start at the mouth because we start by ingesting food. We put food in our mouth. When we put food in our mouth, it triggers a couple of things. It triggers a reflex to chew. I don't know about you, but when I put a lifesaver in my mouth, I often find myself chewing it instead of sucking it, just not thinking about it very much. But there is a reflex to cause chewing. In addition, when food is put in the mouth or smelled or begun to be tasted, we start to make more saliva, and that saliva has enzymes in it that begin to act on the surfaces of those food particles that we're creating as we chew. So in order for the enzymes to work, we're going to have to grind the food, chew the food, masticate the food, and increase the surface area. And then we swallow. Swallowing has 3 phases. Another name for swallowing is deglutition. So that's a medical name. It's not something you're going to hear a whole lot when we are trying to study swallowing. In a patient, we're going to call it a swallowing study, not a deglutition study. But if you hear that word, that's what it means. So the 3 phases of swallowing include a voluntary phase. When you feel that your food has been chewed enough, you use your tongue to push it into the back of your oropharynx. Once the food enters the posterior oropharynx, it triggers the pharyngeal phase of swallowing, which is a series of contractions that move the food out of the mouth and into the esophagus. I'd like to say that it occurs because of swallowing receptor areas in the epithelium of the posterior oropharynx. So we have put the food into the oral cavity, chewed it, and then used our tongue to push it to the pharynx, which is the oral phase. And then the soft palate has to elevate to seal off the nasopharynx, because when you swallow, you don't want the bolus of food or fluid going up into your nasopharynx and out your nose. It's been known to happen. But that's not what's supposed to happen. The soft palate elevates to seal the nasopharynx. I'll tell you that when I was in, 3rd grade, my very first boyfriend, a little boy named Teddy, he broke up with me because I laughed at him when milk came out of his nose in the cafeteria. Alrighty, so in addition to the soft palate elevating, the larynx and the hyoid bone begin to move upwards and forward, and the epiglottis moves backwards and downwards, and that closes off the trachea so that you're not going to get the food that you're swallowing into the wrong tube. Again, it's been known to happen. You stop breathing, and the pharynx shortens, and the bolus moves out of the mouth and into the esophagus. So that was the oropharyngeal phase. Only lasts about a second. It's a very quick process. But once the bolus is in the esophagus, the esophagus begins to contract with peristaltic movements that sequentially constrict behind the bolus, pushing it down the esophagus. Those are the primary movements. You can then have secondary contractions in the esophagus if the primary motions aren't enough to propel the food out of the esophagus through the lower esophageal sphincter or the gastroesophageal sphincter and into the stomach. So the oral phase lasts as long as you chew until you push the food backwards. The oropharyngeal phase lasts about a second, and when everything's moving correctly, it takes about 8 seconds for the food to move down out of the esophagus and into the stomach. This is a drawing to show those phases. So here we have the food in the mouth and being propelled by the tongue into the oropharynx. At this point you can still breathe, right? Because the epiglottis is open and the esophageal sphincter is closed. But then when you're beginning the second phase of swallowing, the pharyngeal phase, the food is propelled backward. The soft palate closes off the nasopharynx, and the epiglottis closes to shut off the trachea, and the upper esophageal begins to open so that the bolus of food can pass into the esophagus. Once it's in the esophagus, the upper esophageal sphincter closes and the bolus is propelled by sequential contractions of circular muscle around the esophagus. In the mouth, we make saliva, and saliva is added to the food. And during the course of a day, that saliva, which contains some digestive enzymes, it contains salivary amylase, and we'll talk about that a little bit more, in salivary lipase. Not a lot of digestion happens in the mouth, but it begins. There is about a liter of saliva secreted a day. In addition, there is a good deal of mucus that is secreted, and there are other substances like digestive enzymes along the course of the early small intestine and from the pancreas's exocrine function. And then, of course, the stomach acid, hydrochloric acid in the stomach, and bicarb solution from the bile ducts and from the pancreas and in the small intestine. So during the course of the day, you make about a liter of saliva, about a liter of pancreatic solutions, about a liter of bile, about a liter and a half of stomach secretions, including stomach acid and the digestive enzymes that are added to that. The small intestine adds close to 2 liters. You can see that the pH is quite different as we move along here. That saliva is relatively neutral, maybe a little acidic. Gastric secretions are very acidic. Pancreatic and bile and small intestine secretions are a little bit alkaline, but they're much higher in quantity than gastric secretions. So these alkaline secretions are able to counteract the acidity of the chyme that has been mixed with the gastric secretions. The Brunner's gland secretes a little, the large intestine secretes a little, and during the course of a day, you secrete about 7 liters of fluid into your alimentary tract. These secretions are controlled, they're in response to stimuli that include food that's ingested and the types of food that's ingested. So if we drink about a liter or 2 liters of fluid a day and we make about 7 liters of fluids a day. We have to absorb all of that fluid back. So the intestines daily absorb between 8 and 9 liters of fluid. None of it's absorbed in the stomach because the mucous membrane of the stomach is not designed for absorption. It's designed for secretion and protection. It doesn't have villi and it has tight junctions between the cells. But when you get to the small intestine, you have structures that are intended to create an enormous area for absorption. So there are folds called Kirkring's folds, and then there are villi or projections of epithelium that are on the epithelial surface of the small intestine. And then each cell on the villus, those cells have a brush border, similar to the brush border that we talked about in the kidneys. Again, massively increasing the surface area of the cell membranes on the epithelial surface of the small intestine. Here we have an illustration of those folds, the folds of Kirkring, and then a close-up of the folds showing that they are covered with a mucosa that consists of villi or finger-like projections into the lumen of the small intestine, and that each of the villi is covered with epithelial cells that have microvilli. So we have just this enormous surface area. The villi contain a blood supply, so an arterial, capillaries, and a venule. Obviously the arterial blood is to supply the oxygen that is required for the very intense metabolic activity going on in the small intestine. The venule is in the portal system and will carry all of the nutrients that have been absorbed from the villus, from all these epithelial cells with their microvilli, in and then be returned through the portal system to the liver. And then there's a lymph vessel called a lacteal, which is crucial in the absorption of fat, that joins up with lymphatic vessels that empty into the thoracic duct. So this is where absorption takes place. Back to the mouth, because we're talking about secretions and then we're talking about absorption. The saliva is produced in several glands that empty into the mouth. The parotid glands are the largest and secrete the largest amount of saliva, which is mostly water. It's a hypotonic aqueous fluid. It has some electrolytes and it has some enzymes to help start digestion. And those enzymes include alpha amylase, which is also called ptyalin, and lingual lipase. In addition, there are glands called submandibular glands and sublingual glands, along with the lymph nodes that tend to be associated with them. And these glands tend to produce fluid with more mucus in it and without the enzymes. And these fluids are to lubricate the food bolus that's being chewed in the mouth. And they all also help with oral hygiene. You can see the parotid gland in front of the ear, the submandibular gland kind of under the mandible, the sublingual gland under the tongue, and lymph nodes associated with them. These are the preauricular lymph nodes. We've gotten to the esophagus, and the esophagus has a couple of kinds of movements. We talked about peristaltic movements. There's primary peristalsis, which is the continuation of the wave that starts when we move food out of the pharynx into the esophagus, continues all the way to the stomach, but that's not always enough to fully propel the bolus. And so there's also secondary peristalsis. And secondary peristalsis results from the stretching of the circular muscle in the esophagus, and when primary peristalsis has not moved the food adequately, secondary peristalsis gets it moved to the stomach. There is a sphincter or tightening, a tonic contraction of circular smooth muscle at the distal end of the esophagus that we call the lower esophageal sphincter, LES, or gastroesophageal sphincter. And as the peristaltic waves are moving towards it, propelling the food, it relaxes, allowing the food through that area of tonically constricted muscle into the stomach. The stomach relaxes as food is entering it to facilitate movement of the food into the stomach and by decreasing pressure in the stomach. The upper esophageal sphincter keeps air out of the esophagus. The lower esophageal sphincter keeps stomach contents out of the esophagus. The lower esophageal sphincter, when it's not functioning properly for any number of reasons, can result in GERD or gastroesophageal reflux disorder or disease. And among the things that can cause gastroesophageal reflux are anything that increases pressure inside the belly, like obesity or pregnancy or even a tight belt. But there are also things that relax the smooth muscle in the lower esophageal sphincter. Some medications can do that and contribute to GERD. When there is damage to any of these three cranial nerves, you can have disorders of swallowing. The fifth cranial nerve is very involved in motion of the tongue. It's the trigeminal nerve. It's the primary motor nerve of the face, but is also involved in swallowing. If the ninth or 10th nerves, which are both involved in swallowing, are paralyzed, we also can have problems with swallowing. Other things that can cause swallowing problems and problems with the esophagus include brain damage. Things like stroke or inflammation can damage the part of the brain stem that helps to control swallowing. Muscle disorders like muscular dystrophy or myasthenia gravis or botulism can cause swallowing problems. And then there is a condition of the esophagus called achalasia, in which there's a very high level of contraction at the lower esophageal smooth muscle that won't relax and allow food to pass through, leading to dilation of the esophagus, sometimes regurgitation. I'm going to show you an image from UpToDate. This is an image of a barium swallow study in which you have a characteristic barium finding of achalasia that's called a bird's beak. So you can see here that this esophagus is massively dilated as he's swallowing the barium. It just accumulates in the esophagus, and very little of it is being allowed through this area of high tonic contraction. This requires a dilation by the gastroenterologist. They put an instrument down there and dilate the lower portion of the esophagus to allow food to pass. Let's talk about the stomach. The stomach has a number of functions, among which are motor functions. I mentioned earlier that there was an exception to the 2 layers of muscle in the gastrointestinal system, and the exception is the stomach. The stomach has 3 layers. It has a longitudinal layer, it has a circular layer, but then interior to the circular layer there is an oblique layer. And this is because the stomach has these particular motor functions. One thing it does is store the food that you eat, so it can actually stretch quite a lot. It can relax and allow a lot of food to enter it. It then mixes the chewed, masticated food that's coming from the esophagus with the gastric secretions to create what we call chyme, spelled with a Y. I think there are a couple places later where it is misspelled as chyme, but it's chyme. And then the third function is that slowly food is emptied from the stomach into the small intestine, but only as quickly as the small intestine is able to receive it. So there are a number of very tight feedback loops involving neural and hormonal signals to regulate the amount of food that is emptied from the lower part of the stomach into the small intestine. You can see that the stomach has different parts. It's a largish organ. The top of it is called the fundus. Where the esophagus enters is the lower esophageal sphincter or the gastroesophageal sphincter. It has a serosa, the three layers of muscle, a submucosa, and a mucosa. We'll talk a little bit more about the mucosa. There is a greater curvature and a lesser curvature. Beneath the fundus or top of the stomach is the body of the stomach. As we move further along in the stomach, it begins to narrow, and it narrows to an area of circular muscle called the pylorus, which is the end of the stomach, and then the pyloric sphincter, which is that tonic circular muscle holding stomach contents into the stomach until it's time for them to be released into the duodenal bulb. As we swallow and the esophagus propels the food into the stomach, it forms sort of concentric circles in the upper portion of the stomach, stretching that stomach wall and causing a reflex that's transmitted back through the vagal nerve to relax the muscle wall, to reduce tone so that more food can be added. There are glands in the stomach that secrete what we call digestive juices, that secrete a number of substances that are then mixed by the muscular motion of the stomach to increase the content of those secretions with the surface area of the food. So the secretions can only affect the food that they come into contact with. They are being secreted from the mucosa, so there have to be these mixing waves that dig into the food contents and move food around to mix it with the secretions to create chyme. If a wave from higher in the stomach, a mixing wave, gets close to the pylorus, the pyloric muscle contracts to close to keep that wave from pushing food into the duodenal bulb unless it's time for that to happen. When there has been no food added to the stomach for several hours, we get rhythmic contractions of stomach muscle that we feel as hunger and hunger pangs. The emptying of the stomach occurs from peristalsis that occurs in the antrum of the stomach. When the antrum pushes the food to the pylorus, we begin to get constrictions that pinch off bits of food, add it to the chyme, and then it eventually gets pushed from the antrum through the pylorus into the duodenal bulb. There is much more circular muscle at the pylorus than there is earlier in the stomach, and we call that muscle the pyloric sphincter. And it's always a little bit contracted to prevent the passage of food until it's mixed enough and until the duodenum is ready for it. And that degree of constriction can increase and decrease under factors, again, that have to do with hormonal and neural signaling. And the rate is regulated by signals that come from both the stomach and the duodenum, but particularly the duodenum. And that's so that chyme does not enter the small intestine at a faster rate than it can be digested and absorbed. If too much chyme and nutrients enter the duodenum from the stomach, when they are exceeding the ability to digest and absorb, we get diarrhea. There's something called dumping syndrome that happens after certain kinds of abdominal surgery, in which food entry into the duodenum is not well controlled, resulting in quick passage because of the high osmotic load and diarrhea. So the gastric factors that promote emptying are an increased volume in the stomach, but also gastrin, which is the hormone that is made in the stomach and enhances the activity of this pyloric pump, the peristaltic movements in the pylorus that move food up against the pyloric sphincter. There are duodenal factors that inhibit emptying, and they include reflexes between the intestine and the stomach, enterogastric reflexes from the duodenum. So distension slows movement, acidity or high osmolality of the chyme, any irritation of the duodenal mucosa, and the presence of peptides and peptones and amino acids breakdown products of protein. And then there's hormonal feedback from the duodenum that we talked about earlier. Gastric secretion is an important topic. There are glands in the stomach. You remember I mentioned that the stomach mucosa is quite different from intestinal mucosa. It does not have villi or folds, it has pits. And those pits lead into something called gastric glands or accentric glands in the antrum body of the stomach. Within those gastric glands are a number of kinds of cells in the epithelium that secrete different substances. There are mucus neck cells that secrete tons of alkaline mucus. The alkaline mucus then accumulates on the epithelial surface to protect this mucosa from the next two substances, which are pepsinogen, which is a enzyme that begins to break down protein, and they are secreted from cells that are called chief cells or peptic cells. And then there are cells that are called parietal cells. Parietal cells secrete large amounts of hydrogen ions and chloride, so hydrochloric acid, but also a substance called intrinsic factor that we'll talk about later that's crucial to the absorption of vitamin B12. And then finally, in the gastric glands, there are cells called G cells. And G cells are endocrine cells that secrete their substances not into the stomach, into the lumen of the gastric gland, but into the extracellular fluid where they are picked up by capillaries. And the G cells make gastrin, and gastrin is an endocrine hormone traveling from those cells in the circulation to other parts of the stomach where they trigger acid production, among other things. Oxyntic glands or gastric glands are in the antrum body, but pyloric glands are in the antrum and the pylorus, and they secrete mucus and gastrin only. They don't secrete hydrochloric acid, or very little, or pepsinogen. So higher in the stomach, there are more gastric glands, oxyntic glands that secrete all of those things. In the distal stomach, we have pyloric glands that have the mucus cells, but they don't have parietal cells to speak of, or chief cells. They have G cells that secrete gastrin. They also secrete somatostatin and ANP from enterochromaffin cells. So there are other kinds of endocrine cells in the stomach. They look similar to oxyntic glands, and they are pits. In addition to the mucus neck cells, there are surface mucus cells. And so mucus and as I said, it's alkaline because it contains bicarb, completely lines the surface mucous membrane of the stomach, which protects it and lubricates it. So it allows food to be moved, and as mixing occurs, it reduces friction to protect the mucous membranes. As I said, it's alkaline, so it protects the mucous membranes from the intensely low pH, the high acidity of the stomach contents, and just any irritation and any contact stimulates secretion of more mucus. Now, gastric secretion occurs in three phases, and I find this really fascinating. Gastric glands begin to secrete in a larger quantity just from the thought of food, or from the sight or smell, or ultimately taste of food. So the first phase is called the cephalic phase, and it results from either thinking about food or seeing it or smelling it, and then certainly tasting it. And it accounts for about 30%, about a third of gastric secretion. Once the food hits the stomach, we get the gastric phase of gastric secretion, which accounts for about 60% of total gastric secretion. But there continues to be some increase from baseline in gastric secretion as food enters the upper part of the small intestine, and that's called the intestinal phase. And it accounts for about 10% of gastric secretion or gastric acid. And the next slide is just a diagram that is showing you some of the events that either stimulate or inhibit these three phases. So the sight or thought of food stimulates the cerebral cortex. You can have a conditioned reflex. You can salivate when you think of, I don't know, buffalo chicken wings. If the taste and smell receptors are stimulated, signals are sent through the hypothalamus, through the brainstem, the hypothalamus and the medulla, through the vagus nerve. So these are parasympathetic signals that are going to increase activity in the stomach. Then the gastric phase is triggered by stomach distension and by some of the chemicals in the stomach, particularly the fact that this food has a higher pH and is going to increase the pH in the stomach, which is going to trigger the G cells to release gastrin, and the gastrin released to the blood is going to travel back to the stomach to cause the release of more hydrochloric acid. And then in the intestinal phase, it's partially digested foods in the duodenum and distension that again trigger gastrin release, this time from G cells in the intestine. And that's a very brief effect. But there are inhibitory effects. Depression tends to inhibit the cephalic phase, and other things that cause loss of appetite. Excessive stomach acidity decreases the secretion of gastrin, which decreases the subsequent production of hydrochloric acid. And then stress that triggers the sympathetic nervous system, the sympathetic arm of the ANS, kind of pushes against the parasympathetic effects, decreasing secretion. And then there are a number of little loops between the intestine and the stomach that also inhibit the intestinal phase, like distension and too much chyme and hypertonic chyme. This slide is showing you a G cell in the gastric gland that is making hydrochloric acid. And it's showing you some of the things that can impact that hydrochloric acid production. There are a number of kinds of receptors on the basilar side of those G cells. There are muscarinic receptors that are M3 receptors that are responding to acetylcholine, parasympathetic input. There are gastrin receptors that are called CCK2 receptors that are responding to gastrin. There are histamine receptors, which, when blocked, act to decrease through secondary messengers, decrease the activity of this pump. And then there are somatostatin receptors. So this is the basilar side with those receptors. The apical side has a pump that exchanges potassium ions for hydrogen ions, bringing potassium into the cell and pumping hydrogen ions out of the cell. And proton pump inhibitors, drugs like omeprazole, directly block that proton pump and prevent or decrease hydrogen ion secretion. There are also chloride channels and potassium channels. So potassium's leaking out and being pumped back in. Chloride is leaking out in a controlled fashion dependent on some secondary messengers. So if you block histamine 2 receptors with drugs like Tagamet, that will decrease stomach acid production. If you take a proton pump inhibitor, you directly inhibit that pump. These are our references. I hope you enjoyed this lecture. We'll be back for more.